that, I will rapidly show you this, how we can predict who eats whom in the food web. So currently I'm professor at the University of Fribourg. I was during 8 years maître assistant in Neuchâtel. It's at that time that I studied the networks. The first picture that you saw, it was the work that we did in Neuchâtel. So it was more on the structure of networks. So I started being interested in web communities, but I was always interested in theory. So I did a postdoc to work on what we call model of species abundance. So in a community, a group of species of the same taxonomic group, I would say, like the birds in the forests or the plants in the meadows. So some are rare, some are abundant, and we are models to explain this abundance, and that's I wanted to work on that. But when I was with, this was in San Diego, at the Scripps Institution of Oceanography, where I started to be interested in food webs. And I worked mostly on the structure of food webs, not on the dynamics. I think my only work on the dynamics is the one on feasibility, where I really looked at what is the dynamic of the network, of a food web. And then I started to be interested in biodiversity and ecosystem functioning when I became professor in Fribourg with experiments. And I first worked with what we called stone wildflower strips. In French, it's jachère florale. So these are compensation areas of farmers. Farmers, they give a percentage of their land to have hedges, trees, or so on, and these what they call wildflower strips. They sow wildflowers. And we worked on that. We manipulated the richness, and we studied. It's extremely complicated, this system. So now I continue with things like that, but with microcosm that I will present. And before I start with what I wanted to show today, we worked with Rudolf Rohr, the model that I will present from here. Christian is here, and we worked on, in fact, it's Christian who started with that. Huh? Here we have a food web, and the idea is to predict if we have or not an interaction, so a blue dot here. And we can use, as we said, the body mass. And what we use is the ratio, the log of the ratio of body mass of the predator divided by the body mass of the prey. That's our explanatory variable. So we can represent the same food webs that we had before. So the ones in blue are here. These are all the zeros. And here we have all the interactions, so the full S by S matrix. And with the log ratio, the log of the ratio of body mass. And here we have the so we can simply have a logistic regression with a quadratic term to make something looks like a Gaussian. And we have that. The predicted number of interactions are all the interactions that are in this cut here. They are here. So that's the observed network, and that's the predicted network. And so this is simple. But then what is interesting is that we can compute the residues of this.
I just have a question. So for instance, if I take an elephant and a mouse, that's not going to work.
Sorry?
If I take an elephant and a mouse, that's not going to work.
So the difference in body size would be, here these are networks that include, where we removed the plants. So an elephant usually eats plants and the mouse eats.
So he's not in the system?
Yes, because the problem, we cannot measure the body size of the plant. So these are part of networks where the plants are excluded. So that's only for the higher part of the food web.
The elephant.
That's a limitation.
The elephant is still there.
The elephant would, no, because if it eats only plants, it will not be here. We exclude the plant, the elephants are out.
But then a lion might eat the elephant, so elephant has
That's that, well, normally not, but that would be, yes, lions would be here, and all the I would say we'll start with the herbivores.
okay.
And everything that goes on.
Elephant is a herbivore.
Yeah.
We start with the, that's true. Elephants would be there because they are herbivores.
But
They
then
would eat nothing. They would be the basal species.
it would be there, but then nobody would eat them.
Absolutely, because
And-
they are outside,
and-
probably outside of the
Right, and then you say it makes sense because the death rate of elephants due to predation is kind of low. Is that what you? And then you justify by saying that the death rate of elephants by predation is kind of low anyway.
Yes, well, here there is no really death rate. It's just, is there an interaction or not? And we know that, well, to have an interaction, if you think at fishes, they can not eat the very small ones. They cannot eat the ones that are too large. There is a range of body size where they can.
Okay.
And that's what we have here, in fact. In fact, we expect that there is a range in body size that can be eaten.
And how do animals like vultures fit in?
So they would be the top predators, and they could not, well, we have no vultures, but they could not eat the very small.
Anything that eats dead animals. And
We would exclude them as well.
that becomes independent of the.
No, that exactly. So there are many interactions that it's really the trophic interactions here between living animals.
Right, thank you.
Yes.
What's the basis of your logarithm?
That's the natural logarithm here. But in ecology, I very often found the base 10, and they don't say.
Yeah, but 20 is a bit much.
So I have also graphs where we see the body size. We studied that's the paper that's the most cited paper, but I'm just a co-author. So we looked at a lot of food webs, and we look at the interaction between the prey and the predator, and we look simply at the body mass of the prey and the body mass of the predator, and this make a big cloud. And the first author who studied that, and it's on base 10 now. And what we find is generally you have to be 100 times larger than your prey.
You have to be what?
The predators are generally 100 times larger than their prey. But it's huge variability. But if we take the trend, it's about that. So and then I'm not sure if it makes sense, but I think yes.
And what about wolves?
Wolf.
Yeah.
But again, it is a general model. So wolf can eat prey that are larger than themselves, of course, if they eat in. So you will have the point in the data.
Yeah, they probably make me.
It will be. It will. So you see the wolf, the elephant. It's part of the variability, so it's part of the extreme in the variability. Okay, perhaps I will stop here. But well, here all the theory on food web was based on body size, and there was a structural model that was really based on body size, the niche model. And when I arrived in this thing, as having worked with birds. When you study birds, you go, well, I was studying forest, and you see that, that all granivorous birds belong to the same family of fringillids. All insectivorous birds, they mostly belong to the same family of the sylvids. And I thought, well, it's clear that phylogeny has an influence on the structure, and it's here that I suggested the model to describe the food web structure, but based on the idea of the nested structure of ecological assemblages that is due to the phylogeny. And here in this study, we rightly said, well, perhaps we can extract what is not explained by extract what is not explained by the body size and it will be measured by the residuals in a way, and we define them as the latent trait of vulnerability and the latent trait of foraging. So that's the fact that the prey is eaten by the predator, or by the trait linked to a predator, and it gives a value what it should eat. And if we make a model with this latent trait, so that was really Christian to add this latent trait. So we add here, we have this matrix of residuals and we separate them in two matrices. It's like a singular value decomposition, in fact. And here, the prediction, you see, that's the observed, that's the prediction. It's really excellent. It's not surprising because we add a lot of parameters in it. But what is interesting is that then we have vulnerability traits and foraging traits that would represent a measure of what is not explained by the body size. And here, yes?
I see VI, delta, and FJ.
VI, delta, FJ.
Yeah,
that's
so
for the predator, that's for the prey.
And delta is what? Delta is an...
These are two this is a vector multiplying a parameter, multiplying, or is it?
It's not. It's just two vectors. It's the inner product of two vectors multiplied by this coefficient delta.
Exactly.
Delta.
It's a latent model variable from statistics.
Yeah, but it's just the inner product of two vectors, right?
Yeah.
Okay.
And they should explain something which is hidden, which is outside, not explained by the body size in the data.
So the point is that we can, in a way, quantify something that explains the interactions, but it is not linked to the body size. And what we found is that these things here are related to the phylogeny. Okay, I stop with that and I start my... Because this is only for the structure, there is no dynamic.
VI, c'est un vecteur.
Okay, effect of temperature change on the functioning of natural community.
Oh, sorry. How in your previous slides? So I'm really sorry, but maybe you don't need to. How many equations did you have an equation for every I and every j?
No, not an. Well, it's a statistical model. No? It's a purely statistical model.
C'est une régression.
Indeed, for every I and every j, we have a parameter. So the number of parameters is then, before for the model was 2, and now it's 2 plus the number of species, in fact. So it's not that surprising. The point is that we can estimate these things.
Yes, you see.
Yeah. Okay.
Well, we can talk about that later.
Yeah, because I mean, I was just trying how many equalities you had when you wrote this. So do you have one for every I and okay. So do you have n square inequalities or where n is the number of species?
Yes, but it is we can discuss this. It's quite technical. It is a model from a latent model.
This model came from...
I think the question is more basic than that.
Yes.
The question is more basic. Maybe, can you just go back to the last slide?
Question of statistics.
It's a question of counting. Yes,
So,
exactly.
so there are n squared inequalities. There are n squared equalities.
Where does J live? It seems to me that you have one such equality for every I and for every J. Am I correct?
You can answer. I have to check again, I have to say.
I think this equation gives a probability that animal I eats animal J.
Yes. Okay.
Yeah.
So you have n squared.
Absolutely.
Yeah.
Okay.
Right, thanks a lot, sorry.
And so there is no summation.
You have understood.
Yeah, there's no solution. Yeah.
Fine.
No, no, it's like that. So, as I said, I started to be interested in what we call in ecology biodiversity, ecosystem functioning. So biodiversity is the number of species, ecosystem functioning. There are different ways that ecosystem function, but the classical measure is productivity. Okay? So the production of matter. And the first studies that showed that, in fact, 20 years ago, it was difficult to convince the public that biodiversity is important, that we should preserve it. We had very few arguments for that. I have to say now, we don't need arguments because I have the impression that it's something that we don't have any more to explain. We have to save biodiversity, it's like that. But at that time we needed arguments to say that. And one argument came from an experiment that started in the 80s that was done by David Tillman. And I will show I should start with showing this one here. Because that was done here, in Cedar Creek. So you see these are, so the ecosystems that I'm talking about now, these are in fact communities of plants. And they saw plants of different species, and they manipulated the number of species, the biodiversity. And what happened in the 80s, there was a drought. So this cost a lot. This was to establish that. It's quite huge, and you have to remove the colonizers. You always have colonizers, and if you want to keep the diversity, you have to remove them. So it's a huge amount of work. So in fact, what happened is a strong drought. Everything died out. It was brown. And here you have the data. Plant species richness before the drought, and the biomass ratio, the biomass in 88, so after the drought and before the drought. And it's quite spectacular because you see that this biomass ratio, so it never went to 1 again, so it's always a bit lower, but clearly in the plots where you have more species, it recovered much better. And then, so that was the first indication that in fact biodiversity is important. The system functions much better, it can recover much faster.
Okay. Sorry, I'm not sure I understand the graphic.
Okay, here we have the plant species richness in 86.
Right, so it's like the number of different plants you have per...
Exactly, they had 9, well, they have here, don't remember what are these values, but...
So 25...
Yes, they add up to 21 species, I guess, originally.
Oh, right, and then you count each...
You take the ratio of the biomass after the drought divided before the drought. So if it is 1, you go back at the same biomass before the drought and after the drought.
Okay.
And we never went to 1, but what we see is that we go to much better, higher value if we have more species than if we have few species. So if there were few species planted, it seems that the system after the drought had really difficulties to recover. If we had more species, after the drought, it recovered much better. Yes?
And what about the ratio, about how many plant species you had after the drought and before the drought?
I think that's the value that... I
Oh,
don't know. I should read again the paper. I should read again.
Because you could have increase in biomass because you have one colonizer or one species that...
The colonizers are excluded. Always excluded.
Well, one species that you already had that really recovers well from the drought, and others that
That's
don't.
a very good question. I will come on that later. Because people, because of that, started to make... I wanted well, I go to the data and then I will come back to this. So the data, so we have Cedar Creek that started again. And you see in 99, they made this experiment with, these are monocultures. We have two species together for 8 or 16. And this is the biomass, average biomass, and you see that, oh, okay, this is the log scale, base 2, and we see that it increases. The biomass produced, or I would say the productivity, you know, because it's always, so they cut the grass after some time. So here in this Jena experiment, it's still ongoing, and 2 times a year they cut everything. And also they remove all colonizers. And they look at the productivity, the relationship. You see here you have several years. So when they started the experiment, there was some relationship between, so 1, 2, 4, 8, 16. And we see that in 2000, it seems that with time, the effect became stronger. The effect of diversity becomes stronger. So here they made a big experiment in Europe, in several countries, and they had the same system. And here you have for the graphs for every country, you have the value of the biomass of the individual plots, so there is a lot of variability. But you see that in general, it increases. So they went up to 32 for some places. There is only one that did not increase, it was in Greece, but it's this kind of thing. But there was a plot in Switzerland. I think this is the one in Switzerland. There was some in England. And we see that in general, when we have higher diversity, globally, we have more productivity. Models for that. Tillman, who was the initiator of this experiment, proposed some models. So competition for one resource, competition for 2 resources, a generalized niche model. So it looks similar to what we did, and I will explain in a moment. But you see, theoretically, you would expect that with the species richness and the total biomass, you should increase. So it predicts this increase with competition model. And here you have a general model where this is all the resources. You can imagine a niche space, 2-dimension, I don't know, the gradient of nitrate, the gradient of phosphate or whatever. And each species consumes a part, well, it makes best in some ratio of these 2 things. So you can draw species randomly as circles, and you can give the radius and so on. And you see that basically if they are more, they will eat more resources so they can do that. The problem with the experiment is exactly what you said. How to interpret that? There was a big fight about ecologists yelling at each other sometimes. And in 2001, Michel Laurent, who is a Belgian ecologist, quite famous, and he made a conference where he put all people together and they came on this. So we have a species pool from which we assemble a community. Now, why do we have higher productivity? It can come from different processes. They said it's because we have more species, so we can use the resources available more fully because we complement each other to use all resources. That's the idea of complementarity among species with different traits. In the experiment, it was found that when we add the legumes have special roots where we have bacteria that fix the nitrate. When we have this species, it increases the productivity. So if we have different functional groups, like it is maybe perhaps the legumes, then if we have groups that do things differently, they can also have an effect. not only at the species level, but also perhaps at the group. And the critique of the people who said, well, in fact, this thing here, it's tautological because if you have more species, you increase the probability to have one species that do very well. So it's just what they call selection process. So if you have more species, you are more likely to select the good species. It can also happen. It can also. It's not. These things are in fact not exclusive. This can also happen, and all these things can contribute to the higher productivity.
Since you measure the biomass of every species, if it was the case, it will only increase the biomass of the species. How do you say that? Singular species.
Mm-hmm.
The species which is very good, no?
Yes, because
but
in fact
all species were increasing, so.
You take the median, so it should.
Sorry?
You take the median, so you say what you say is correct. And if it's just one species that is, but maybe one species is really productive for a lot of other ones.
Ah, you mean one has a good influence on? Okay.
I think that's the point.
I'm not sure to have understood the question. Thank you. So the point is that Laurent and Hector proposed a statistical method, partitioning of the method to distinguish between, I don't want to enter in the details, but to the selection effect and to this complementarity effect. And they reanalyze, because they have you need the monoculture to do that. You need to know what are the good ones when they are alone. And then you can separate this effect in this complementarity and selection. This is the data for the experiment in Europe. And what they found is that there is no evidence on effect of selection. So it's not the fact of including the plant that there's a high productivity when alone that will create this increase, but it's really the complementarity effect.
How did they check
So in general, sorry?
How did they check this?
What?
How did they check for the selection effect?
So in fact, it's a statistical model where you look at the residuals, again with a covariance thing in the residuals. And well, I can show the paper.
Okay, so
So
it's just a statistical procedure. They don't do something on the field where they
no.
take out other.
Exactly, but to be able to do that, you need to have the monocultures.
Okay, I see.
Because otherwise you will not be able to. And it's also the reason why you don't have the value 1 here that existed in the. Okay, that was the background. Now we know that biodiversity is good to make communities perhaps more happy. I don't know. I think so. When you look at the data that's something that we did. We look at the Jena experiments through time, and we realized that at the start of the experiment, it seems that the selection effect is, we did not use the same approach. It seems that the selection effect is weak, and then with time it becomes stronger. It's in link with what I showed before. So you have the impression that at the start of the experiment, the strong one were taking the space, and then little by little, they decrease in abundance, and the density becomes more equal. So
You said something about removing the colonizers in the beginning.
that's because we did the same experiments in these wildflower strips. That thing we did not do because it's too demanding in time. And also we did not do it because it makes a perturbation on the rest of the system. And we wanted to know what was the effect on the higher trophic levels, so we decided not to do that.
So it's much more interesting not to do it, right?
Because
you would think
I would need 100 students to do it.
So what you're saying is that you were removing the colonizer because you were afraid that one species would just kill everything else?
Well, it's to keep the original number of species. You can have extinction. You can have a species that disappears. But the idea is to try to keep the community with the species that you decided to put together. So that's only that.
Oh, so the colonizer were external thing that were
Yes,
not.
colonizers are always external species
okay.
that would come. So if a colonizer come that belong to the species in the mixture, you will not.
I see, okay.
The colonizers are species that.
That you didn't want to include in your
Yes,
your
yes.
study.
So you will increase your diversity, and well, it's to keep in the experimental, the nice experimental setting. So now the big question is, that interests us is what happens if we add temperature on that. And it's the reason I'm here. So I know that with plants there are some experiments, notably in constants of a colleague. But you imagine when you have to warm drugs that it's very difficult. And for this reason, I changed my system. I stopped working with these wildflower strips, but to work with microcosm that I will explain in a moment. So these are natural microcosm that contains bacteria and protists. And we did the experiment with protists because we can keep them in incubators where we can incubate. But with the experiment, it would do a prediction. And the prediction comes from a model that was developed by Fribourg, so the first of I have a picture of him. So this is again a kind of Lotka-Volterra model where we have consumers consuming only one resource. The whole theory that I will show works also for more than one resource. It works also for resources that are not a quantity, but that can be that you can represent quantitatively along a niche axis or whatever. So it's quite general. So should I explain that? Yes, I think. So here, that's the consumer when it is alone. This thing here will disappear. This thing here, it will only stay with alpha II and Ci. And here we have minus m. M is the mortality rate. When the consumer is alone, in fact, he will go to 0. Okay. That's the mortality rate of the consumer. Here, that's the interaction with the resource. So that's in fact, the attack rate of the resource. Okay. It's the attack rate. And this is the efficiency. I don't remember what sign you use, but that's in ecology what we say it is efficiency. That's in fact the transformation of biomass of resource into biomass, if you want. But here it's the effect of a change in resource in the growth rate of the consumer. It's just a transformation factor. So the efficiency. And now here we have the interaction between the consumers. Because the other consumers may have an effect on this one. We may have a positive or negative effect.
This is the random matrix A from this morning. Is equivalent to the last model we saw?
In our model here, in fact, we separated everything that was due to eating the resource with, well, you will see in a moment, with the interactions that are everything else than the resource consumption. We separate these two things. Usually, it was never done before, but I think it's useful to do it. Because in fact, we know that the attack rate, we know that the mortality rate, we know that the growth rate of the resource, here it's the same thing. Here it's a mortality rate because alone it cannot persist. And here it's a growth rate because we assume that the resource will increase. So these things depend on the interaction. Okay. Now, what we will do, here we have the resource. And if you look at the resource, so it increases. Here, this is the intraspecific competition for the resource. In fact, it's if we have only that, it's the logistic model. Okay. So it's R minus alpha times R squared. And if you heard about the carrying capacity, you use that in the talk? I don't remember.
No,
Never, huh?
I said just a
It's
kappa,
something-
not a constant kappa I.
So the equilibrium density, if you exclude the interactions here, it would be R divided by alpha. So it would be the growth rate divided by the intraspecific competition rate. In ecology, we very often, so perhaps I should use that. You have I can use this one. So the logistic equation. You can move it.
Non, vaut mieux.
You
là.
don't like it?
Non,
I don't
I
like
can't.
it. Ah, yes, me too. So that's the logistic equation. And it's like a sigmoid growth. So I guess it's clear. And in ecology, very often, because we will use that in fact, we use it in the K formulation. We can write it like that. Or n, K minus n divided by K. It's the same thing. Ecologists often did not realize that, in fact, in this formulation, you may think that K and R are independent, but they are not, because K is equal to R divided by alpha. So, in fact, R and alpha are thought to be independent, should be independent, though we just made an experiment where with chemostat where, in fact, with a batch where they are not, in fact,
Is there-
parentheses. But we assume that R and alpha are independent. Okay? Yes?
Sorry, is there an n missing in the second equation?
The M is only for the consumer.
N.
N factor of your second line. One n is missing.
Thank you. Yeah, of course. Rn. Thank you. Okay. So we see that it's the same thing that we have here. And now its biomass is reduced because it is eaten by this one. So we place this thing here. And, okay? That's the sum of the effect of the consumer. Now the idea is to reduce this model for only one equation for the consumer. And that was done by assuming that the resource has a very high growth rate and, that in other words, that it reproduces fast enough to always go to his equilibrium density. So we can assume that we can get rid of this thing by measuring its equilibrium density, and then we can, and assuming that by the fact that it has a high reproductive rate, it will always go to that, and then we can use this equilibrium density here. So that's what we can do. And you see the gamma interaction between consumer A and J other than exploitative competition. Here we don't have the exploitative competition yet. It's just the attack rate of each species. We don't have competition because we have just one thing for the resource here and for the consumers. The consumers attack the resource differently. And when we take care only of the consumers, then they will compete for it. So we can assume again that Okay. So these things, the dynamic is much faster. So if we have to find the equilibrium, we have to put that to 0. You agree, I guess? And then we obtain that R star 1 divided by alpha R minus the sum of the continuum. Okay? And then that we can plug inside this one here. And it becomes a bit more complicated by rearranging the term. But we have this thing here is the growth rate of the consumer. Alone it would die. And here it's what is gaining through the fact that it is eating the consumer. Okay? And now, because we have two interactions here, we have, it's here that you mentioned the matrix that...
yes, it's exactly the sum of the last slide.
So here we have the interaction that is non-trophic plus the interaction that is trophic. So we have the effect of, it's always the effect of J on effect of E, if you want. So we have efficiency of A, the attack rate of A, the attack rate of J, and always divided by this alpha R, that is the intraspecific competition term for the resource. So we can represent this like that. And from that, we can formalize this in the RK notation. Because it's useful to have the K to come to this model for, in fact, I'm not sure I said that, but the first step is to obtain a model for the BF relationship. This thing that we saw before, I want a model. So, okay, we can formalize that in this way. And here we have the growth rate of that is equal to that. We use RA instead of this whole thing here, the mortality minus the mortality plus the increase due to the consumption of the resource. The K is by definition defined as RA divided by the intraspecific competition for the species with itself. And it contains the things that is non-trophic plus the thing that is trophic. And here we have this RA that was before. Now we have we call this the standardized interaction coefficient because it is standardized, that was the one that we had before. It is standardized by the coefficient, the interaction coefficient for the species with itself. It's the same thing as this one, you see. So it's not an absolute interaction coefficient between two species. It is standardized by your own intraspecific coefficient. Yes?
Could you just recall what trophic means?
Trophic, it's eating the resource.
Okay.
And non-trophic, it's everything else. It may be, in fact, a facilitation as well in the gamma. So now that we have that, we can solve this thing at equilibrium. And the system at equilibrium, K1, oops, you see, we take care of it on this one, and we put an equal here. It's not more.
Yes, but it's not the true equilibrium. Huh?
Sorry?
It's not the true equilibrium.
Absolutely.
It's a highly simplified version where you suppose that they are all coefficients are the same.
Exactly.
Replace each coefficient by the mean.
Well, here this would be the true equilibrium. This one here. It's correct?
Yes, this is the right equation, but after the solution.
Absolutely. So here, the problem is that how to solve that? It's difficult. And also, we want a theory that links the number of species, S. We need the number of an expression for the number of species, and for the productivity.
Sorry, I got kind of lost. What's KI?
So K is the carrying capacity. That would be the sum of the carrying capacity of all your species.
But that's a number that doesn't vary over time.
Sorry?
That's something that doesn't vary over time. The CIs vary over time.
No, you're right. They do not vary over time. Here we are at equilibrium. So we attain K. Well, I think for me the most difficult part is this one, but you can convince yourself that starting with this one here and going here, you obtain the same expression.
So, no, but to me, K was a parameter, so a real number.
But the say start does not depend over time
So
in the equation, so you have only time independent quantity.
C star is the equilibrium
Yeah.
point of the CI T independent of time. Okay.
Sorry,
Okay.
I was not clear with that. I think in ecology it's so clear that K is the equilibrium density that...
Even the linear system of the
and K is the equilibrium density when the species is alone.
Okay.
Okay.
Right. Yeah.
That you can measure with monoculture.
Yeah. Okay. All right.
That's the point. Here you need again the... No, it's good. It's perfect. And you see, in fact, these are the equilibrium density of all the species where they are in the mixture.
Right.
And it's 1 times C1 because that's, in fact, the intraspecific competition with itself. And these are the alpha AJ that I defined before, the standardized coefficient of interaction that you have here with all the species.
Yeah.
And then you have an expression that would link the sum of all species in their own carrying capacity with this value here. Of course, if you imagine an experiment where you put all this together, it's impossible to attain that because the space is limited and so on. In fact, we can solve this thing, and we have an expression for the K, we have an expression for the alpha, we have an expression for the C, and now we can assume, as you said, we don't know the alpha H. But we can assume that they individually, we could assume that they are almost all the same, or not too different, and we could replace them by their mean. It would work if we don't have, for example, cases where we have all species with a very high biomass having, for example, a very small alpha Aj. So, in that case, we can write that. We can define rho, the effective interspecific interaction, as the average of all this value. And in fact, we can see that if we look at that, how many values like that do we have? We have s minus 1 because we have the 1 here values, and it's simply 1 divided by s minus 1 and the sum of all these things. Or we can write the sum of all these things is proportional to 1 plus s minus 1 times rho. And now we can write a BF model by saying that the sum of the abundance in polyculture, the sum of the abundances of all species in polyculture, divided by the average value in monoculture. This is important. It's called the relative biomass. If you want to compare, it was already used to compare different studies. You see, if you have different species, they may go at K that are completely different. Or sorry, not at K, but at total biomass that is very different, it depends on the species. If you want to compare them, a good way to do that is to obtain your total biomass in polyculture and to divide it by the average carrying capacity of each species in monoculture. So that's the polyculture divided by the monoculture. and we assume that it scales. It again, no, this is exact, but then we can write only that. It scales with s divided by 1 plus s minus 1 times rho. And that's a simple model that goes to an asymptote. No, we thought at that time that
what goes to an asymptote?
This thing here. If we have 1,
But to me, that doesn't vary over time.
no, these are at the end of your experiment. You are at the end of the experiment. You have your monocultures. You measure the carrying capacity. You have your polycultures. You measure the sum of, so you measure the total biomass, which is the sum of your individual biomass.
Yeah.
You make the ratio of this value by that. And that's what you measure at the end of the experiment. We are at equilibrium. The dynamic was before.
Okay.
And now you see that, in fact, if rho equal 1, we have 1. It means that there is no there is no benefit of being in polyculture. We would say the same. Now, an interesting thing is that, well, I will pass very rapidly. When Rudolf, our colleague, came with that, I thought, wow, we have the equation for the BF relationship. And I was thinking, well, we call this the funny name for that. And then we realized that, well, first thing, before I talked about that, empirical support. So this is from a meta-analysis of about 50 different studies where they manipulated the species richness that we have here. And here we have exactly this thing. That's what we have before. They call this ys divided by the average of m. So that's exactly that's the total biomass divided by the average in monoculture. And what they found, sometimes you see it's negative, but it's quite rare. So they separated the aquatic and the terrestrial system. In black, it's the aquatic system. But in general, we find this thing. And they say, well, we don't know what is the model. They fitted three models, a linear, a logarithmic one, and a Michaelis-Menten equation. That's what they fitted. Y equal Y max times S, K plus S. I guess you are familiar with this thing? This beautiful equation, Michaelis-Menten.
Not.
Menten is what's the you should read the life of Menten because she's a female scientist in the early 19th century. It's remarkable. This is the asymptote. That would y max. And if you have y max divided by 2, so it's in the cinetic of an enzymatic cinetic. This
Yeah, I think I've seen
and
that.
then you have this thing here. It's the half saturation constant that is called here. Really strangely, they call this K. Don't ask me why they use K because it's stupid. It's carrying capacity. No. So in fact, in our model, we have one parameter. They fitted a two-parameter equation to this, which is stupid. Which is, well, look at the values that they obtain. They obtain in general that, so, in fact, I can represent this equation in the, not using Imax and K, but using the rho that we have. Rho is like the competition, the total competition. 1 divided by rho would be a measure of the complementarity. We can see the competition and the complementarity like the two sides of a coin. They are not independent of each other. So if we have high value of rho, we have low value of complementarity. Okay. And the idea is that we can think of this as a complement to this Y max as 1 divided by rho as a measure of complementarity. If it is high, we will go to high value of this relative biomass. And now this term here, we see that it's just 1 minus 1. So K, in fact, should be Y max minus 1. When you look at the parameter estimates, you see here 2, and this one is 2 minus 1. And you see here it is at 2 point something, and this one has exactly a distance of 1 between 2. In fact, they could have saved one parameter. And they would have got even a stronger statistical support for this relationship. But you see it as empirical support. And we even can have negative values, a negative point here. It's just when the competition is so strong between the species here that in fact when they are together, they do less well. So meaning that the rho here is larger than 1. So the first model that I showed to you, it's impossible to have negative relationship, a negative relationship between biodiversity and ecosystem functioning, to a point that Tillman said, so the author said it's impossible. But no, it's really possible, it was even fun. So then we thought it was the only time it was derived. In fact, it's Christian who told me, this equation, you find it in this book. I was totally amazed. I can remember that having read this book, there is one equation among 1,000 that is the same as that we had. And I check, and in fact, indeed, it's Axel Rosberg in his book. He used this equation here. It's the same one as we had, but we have to rearrange it, change the terms. So put the K to the left. And he used it as a biodiversity ecosystem function. He was the first one to do that. It was also derived funnily by Cardinale in another thing. So again, you have to put the K here. And it was even derived in the 70s by van der Meer. He did not show how to derive it. He immediately gave that. Here we have K divided by N. So you need to put a 1 here. And here he added this covariance term that he said, well, I have no idea how to measure it, so I will disregard it. That would be the fact that we have N associated with special value of N associated with special value of the alpha. And the last one that I want to show, because it's really interesting, it's Hugo Fort. He derived in a simpler way than what I showed. It is exactly the same equation. Here we have the number of species, and here we have the interaction term. And what is nice, he looked at the data. These are the data. For example, this is the biodepth experiment. This is the Jena experiment. And these are simpler experiments, where we have the number of species here. And from that, he looked at pairwise plots. When we have 2 species. When we have 2 species, we can measure the alpha AJ. We have only the 2 species, so we can estimate it. And he estimated them, and here we have the percentage values of this alpha AJ that he has for these experiments. When we have very few species, it's possible to have all of them. When we have more, it starts to be difficult. You see, when we have 32 species, we have 32 times 31 divided by 2 experiments, so it's very difficult. So we have only 3% of the cases with that. But then he asked himself, if I use this pairwise interaction, can I predict the total biomass using the equation that we have before? And that's the expected, and that's the experimental one, and that's the theoretical biomass, relative biomass. And if you make the plot of that, I will stop.
I don't know how much of a crime it is here to arrive late at lunch, but I think that...
I'm at the end.
Okay, great, thank you.
That's basically the last one. But you see, we have a quite good way. Ah, no, that's not the last one because I did not talk about temperature. I will talk about temperature later.
Yeah, this is
Well,
afternoon.
but in fact, temperature, you see, well, I would need 5 minutes.
I think, I don't know how much of, you think it's fine to arrive 5 minutes late for lunch or not?
Yeah, but not more than 5.
Okay. So, temperature, it affects the parameters. It affects them. This is what we call a temporal performance curve. In our model, we only deal with this part here, the suboptimal part, that we can describe with Arrhenius equation, exponential. So with temperature, the value increases like that, okay? So if we do that, our values were that, and now this A is this thing here, and we change it with this one, okay? So they increase with temperature. Okay, the attack rate, that was experimentally shown that it increases with temperature. For protists, we use these values. If the temperature increases, the attack rate increases. Higher, so my metabolism works stronger, I'm faster at grasping the resource. So the attack rate increases. If the attack rate increases from 12 to 30 degrees, in fact, it's the interspecific coefficient. It's the coefficient that I showed before. It's this thing here will also increase. Here, I don't know why I called it niche overlap. It's a bit strange. But, so the interaction will be stronger. It makes total sense. You know, if you have the same resource, you have higher metabolism, your attack rate is higher, the interaction will be high, will be stronger. If the interspecific niche overlap increases, so that is our value for 32 species. And if we put that here, we measure the relative abundance in monoculture. Of course, it will be 1 by definition, in pairs, in free species, and so on. We see that the BEF relationship will go from year to year, so it should decrease. If we make the experiment with protists, so that I will not explain. If we make experiment with protists, these are species that we find inside the leaves of this carnivorous plant that we have in Switzerland at some places. We have 2 sites, one close to Lausanne and one in close to, above Montreux. We isolated 6 protozoan species that eat bacteria. We have monoculture of them, and then we can put them alone in pairs and so on. And we manipulated in incubators the temperature. So we increase by 5%, and we increase by 5 degrees, sorry, not percent, 5 degrees, but we increase the day-night difference in temperature. So we increase the variability. And if we the result in the cold site, we have in fact something special. Here we have facilitation. We had a biomass, so the relative biomass that was higher than what they would do if they were all in monoculture. We don't know what is the mechanism. It's really special, but something happened that they did extremely well. And when we increase the number of species, you see the competition becomes stronger. But here, it was quite amazing. So we have 1, 2, 4, and 6 species. And then, if we increase the temperature by 5 degrees, you see there is and here we increase also the variability, there is not much difference. But for the warm side, we start at higher temperature. So we have a positive relationship at normal temperature. We increase the temperature, it goes here. And if we decrease, we have extinctions. We have no points here. So it strongly decreases and we have extinction. It's not good news for AEF because temperature would tend to decrease the effect of the BF relationship.